Okay, it's my pleasure to introduce Allie. She's a master's student at Mississippi State University's College of Veterinary Medicine. With her previous job at the USDA and her current research, she's spent enough time with catfish. She finds it difficult to eat them. Having always held a passion for animals, she graduated from MSU with a bachelor's degree in wildlife fisheries and aquaculture. Outside of classes in the lab, she enjoys reading, watching horror movies, and spending time with her array of pets, and is thinking of taking up aquascaping. Got a lot going on there, Allie. Let's hear about the research.
Okay, so I'm Allie, and today I'm going to be talking about some of the research that we've done so far for my master's project, which has been looking at the effects of microcystin LR on channel catfish susceptibility to microbial pathogens. First, I just wanted to briefly introduce these two pathogens. These two bacterial species that we're working with are Aeromonas hydrophila, which I have a graph on the right, and Edwardsia piscicata, which is on the left. Both of these bacteria thrive in temperatures between 18 to 29 degrees Celsius, which coincides with the prevalence during the summer months. And this is around the same time that cyanobacterial blooms are very common. So cyanobacterial blooms have substantially increased in freshwater systems. This can be from eutrophication from both urban and agricultural practices, and from climate change, which has resulted in higher summer water temperatures. With the increase of these cyanobacterial blooms, there's also been an increase in toxins produced by them, which can lead to poisonings. So there's an expected poisoning events are expected to increase. And there have been reported cases in humans, domesticated animals, and in wildlife. And out of the toxins that are produced by cyanobacteria, microcystins are among the most common. So microcystins are cyclic heptapeptide hepatotoxins, and they're known for their ability to harm the liver. Out of these microcystins, microcystin LR is the most studied form, and it's recognized as the most harmful and most encountered variant. The primary target of microcystin LR is the liver, and then the secondary has been found to be the gonads. For our research, it's been, in previous cases, it's been unfortunate to be able to pinpoint a clear identifiable predisposing event in bacterial disease outbreaks and catfish grow-out systems, because these older fish should have a natural immunity in place and the usual stressors are not present. Therefore, being able to identify a possible predisposing factor could lead to management practices. For our first objective, we wanted to evaluate the effects of a sublethal microcystin LR dosage exposure on the channel catfish physiology. And we also wanted to evaluate some diagnostic tools that can be used in the future. And our second objective was to evaluate the effect of, again, a sublethal exposure of microcystin LR on channel catfish when they're also challenged with the bacterial pathogens Aeromonas hydrophila and Edwardsia piscicata. We believe that exposure to microcystin LR predisposes channel catfish to Aeromonas hydrophila and Edwardsia piscicata to cause diseases. So for our first objective, we conducted a 6-day trial using 10 tanks. Whenever we handled or injected fish, they were always anesthetized with MS-222. And for this trial, we had 2 treatment groups. First, the microcystin fish that were IP injected with the 500 parts per billion body weight dosage of microcystin LR that was dissolved in saline. And then our control fish were IP injected with sterile saline. For our sample collection, fish were sacrificed using MS-222, and we had sampling periods that ranged from 0 to 144 hours post-injection. During each sampling period, we took liver and serum samples that were then stored at -80 degrees Celsius. We also took gill, liver, kidney, and spleen samples that were then fixed in 10% buffered formalin for histology and immunohistochemistry. And during each sampling period, we would observe the stomach and the lower intestines for evidence of digestion or ingestion of feed to evaluate feeding behavior. So regarding feeding behavior, our control fish throughout the trial continued to feed as normal, and they had ingesta in their lower intestines. On the other hand, by 48 hours post-injection, the treatment fish had reduced feeding. Some of them did have feed in their stomach, but none of it had migrated to the intestines while it did in the controls. And by 64 hours post-injection and all the way to the end, the treatment fish did not consume their feed. So to evaluate the amount of free microcystin within the liver, I took methanol extracts, ran them through a C18 column for cleanup, and then I used an Abraxas microcystins ADA kit. So we evaluated 4 different things for serum chemistry. We looked at aspartate aminotransferase, alanine aminotransferase, alkaline phosphatase, and bilirubin. And we looked at all of these because elevated levels are indicators of liver damage. And these were all conducted by the diagnostic lab here at the vet school. So we did not see any significant trends in ALP and bilirubin, so I did not list the graphs here. This top graph shows the amount of free microcystin in the liver per hour, which is the dark line, and the ALT within a treatment fish per hour, which is that light gray line. So we saw a significant difference. Well, in regards to free microcystin, we saw a significant difference in our treatment at 96 hours post-injection, where we had a mean OD of 221 in comparison to all of the control fish at all hours. In our ALT treatment fish, the gray line, and I have indicated by the arrow where we had our greatest significant spike, which was at 48 hours, and that average was 129.40 units per liter, which then would decrease into 144 hours post-injection. And in this bottom graph, I have shown AST for both controls and treatment fish, in which we saw the largest spike in treatment fish at 24 hours, which was 214.8 units per liter, which then significantly dropped again by 48 hours and continued to decrease to the end of the study. So histology and immunohistochemistry were performed on the fixed tissues to evaluate the pathology and the presence of microcystin within. Our histology samples were processed through the diagnostic lab, and they were H&E stains. For immunohistochemistry, this is something we're still working on, but our primary antibody is the microcystin LR monoclonal antibody, and we used horseradish peroxidase as our secondary antibody. So here are, on the left, we have a liver sample from a control fish, and on the right, we have one from 48 hours post-injection. This one on the left will show the normal vacuolated hepatocytes that form almost a triangle shape around the bile ducts. And on the right side, we can see there is noticeable individualization or rounding, shrinkage of the cells, and the dilated sinusoids. This next slide is images of a spleen. Again, the one on the left is our control, and the one on the right is a treatment fish. This one on the left will show normal looking cells, while on the right, we can see the spleen displays necrosis, which is shown by the moth-eaten appearance in the periarteriolar lymphoid sheath. Here I have some immunohistochemistry results. On the left, we have a control fish, and on the right, we have a fish from 48 hours. So far, we have seen that the microcystin positive staining in the liver is more widespread in the early hours, so around 6 hours, and then through 24 to 48 hours, we start to see the staining more concentrated. And we can also see on this right image, there is the vacuolization of cells, which is also seen in histology. And we are still looking through our 96-hour and our 144-hour post-injection fish. So for our first bacterial trial, we looked at Aeromonas hydrophila. We had 4 different treatment groups, our group 1 being the control, group 2 were bacteria only, group 3 the bacteria and the toxin, and group 4 toxin only. And this trial was just a mortality trial, so it continued until there were no more for 48 hours. To prepare the bacteria, I first took a stock solution of Aeromonas hydrophila and I streaked it onto a TSA plate for isolation. This was then incubated for 24 hours at 30 degrees Celsius. I then took an isolate colony and suspended it in TSB, and that was placed in a shaking incubator overnight. This stock suspension was then read the next day at 600 nanometers, and it had an OD of 1.207, which I then serodiluted on the TSA plates to be able to calculate the CFU. And for this trial, the CFU that we used was 3.1 times 10 to the 9th per mil. For our inoculation protocol, as always, fish were anesthetized with MS222. For our control group, fish received an IP injection of saline. They received an adipose fin clip, and they recovered in 2 liters of static water. Group 2 is our bacteria group. They received saline, the adipose clip, and then they recovered in 2 liters of water that also had 100 mils of the bacteria. Group 3 received a 500 parts per billion body weight dosage IP injection of microcystin, the adipose fin clip, and again recovered with the water and bacteria. And group 4 only received the microcystin, the fin clip, and they recovered in the water without any bacteria. And in this trial, after 2 hours, we had resumed the water flow in the tanks. To sample these fish, I would check them 3 to 4 times daily. Every time there was a mortality, they were counted and removed. And then these fish were necropsied and the posterior kidney was streaked on TSA plates. And for this trial, mortalities after 48 hours was having to end at 168 hours post-injection. So we did see that microcystin increased the susceptibility to Aeromonas hydrophila. And the orange and the green lines was our control on microcystin. There was no mortalities. And in our bacteria-only group, this blue line, we did lose most of those fish by 18 hours post-infection, which we had lost 18.4% of our fish. We lost another by 22, and then the last one was at 114 hours, which we ended up having 77.6 of these fish surviving, or had a 22.4% mortality. On the other hand, we had a total of 67.3% mortality in the bacteria and toxin fish, which again, most of our fish were lost by 18 hours post-injection, and we had lost 26.5 by then. Our last mortality in these fish was at 67 hours post-injection, which had left only 32.7 of the fish surviving. The next bacterial challenge focused on Edwardsia piscicida. Again, we had these 4 treatment groups with our control bacteria and toxin only. And during this challenge, we sampled 5 fish from both the control group and the toxin group at 24 hours to be able to make sure we can get serum and histology samples. And again, like the previous trial, this one continued until there were no mortalities for 48 hours. So to be very, the preparation of this bacteria was very similar to Aeromonas hydrophila, except that after I streaked it onto a TSA plate, it had to incubate for 48 hours instead of 24. Then after this, again, an isolate was placed in TSB and into a shaking incubator. I read the stock at 600 nanometers, and it had an OD of 1.191, which I then serodiluted 1 to 1,000 in saline, and that was what was used. And the CFU for that was 3 times 10 to the 5th per mil. So for this inoculation protocol, everything, if it was introduced to something, would be through IP injection. So our group first group, our control group, they got 2 IP injections of saline and no bacteria. Group 2 got an IP injection of saline, and then they received 100 microliters of the bacteria. Group 4 received the body weight dosage of microcystin and 100 microliters of the bacteria. And then group 4 received only the toxin and no bacteria. Again, I would check the fish 3 to 4 times a day. These mortalities, again, were counted and removed. And for each sampling period, up to 3 of the mortalities were from each tank were necropsied. So again, I took a PK streak on the TSA. I took liver samples, and if the mortality was fresh enough, I would take samples for histology. And in this trial, it ended after 108 hours post-infection. So interesting enough, throughout the trial, we didn't have any mortalities in any of the groups that were just toxin or just bacteria. On the other hand, shown again by this red line, by 39 hours post-injection, we had lost 64% of the microcystin and bacteria fish. And by 45 hours post-injection, we had lost 70. And the mortalities of this group ended at 65 hours post-injection. And by the end of the trial, we only had 3 of 50 fish left, meaning we had a 6% survivability rate or a 94% mortality. So just to summarize everything I've gone over, we found that sublethal exposure of microcystin LR leads to persistence up to 6 days. It induces liver and spleen pathology, elevates ALT and AST levels, and it causes anorexia. In our challenge experiments, we see a significant increase in mortality from Ehrlichiosis hydrophila and Edwardsia piscicida infections in fish that were also exposed to MCLR. And so to conclude, sublethal microcystin LR exposure alters channel catfish homeostasis, and it increases their predisposition to bacterial diseases, meaning that controlling cyanobacterial blooms may decrease disease outbreaks in the summer. I'd like to thank Dr. Larry Hansen, my PI, Dr. Peter Hansen, the co-PI in my committee, Lori Ford for helping with the dosing studies, Dr. Beth Peterman for helping me with the immunohistochemistry, Mark Peterman for doing some of the preliminary Ehrlichiosis hydrophila mortality data and the growth curve, and Heather Cunningham and Nicole McBrayer of the diagnostic services for doing serum chemistry. And this project was supported by Agricultural and Food Research Initiative from the USDA National Institute of Food and Agriculture. Thank you.
All right, great job, Allie. Interesting project.
Thank you.
Folks, if you've got a question, feel free to drop it into the chat box, or you can unmute yourself. And we'll start off with a question that was sent to me from Dave Groman here. He's curious on the justification for an IP challenge rather than a bath challenge.
For the microcystin or for the bacteria?
Both. How about?
I think that was more to have it controlled, to make sure they were actually receiving that amount of the dosage and it can be more focused. I think we had contemplated on doing immersion for the piscicida, but there had been some struggle before with actually getting it to take within the fish through that way. So I don't know, we decided to do IP for those.
Okay. Yeah, hey, Allie, great talk. And I'll follow up Dave's question with kind of that same line of reasoning. Had you guys considered even looking at an immersion challenge? By and large, it's very, very different. We've never been successful in an immersion challenge with piscicida in catfish, especially channel catfish. And so I was wondering if you guys had considered maybe we could do it if we had, I guess I'm curious whether or not you guys considered adding that as a wrinkle to this work.
So we did some preliminary stuff where we actually did intramuscular and IP. We just want to see if there is different ways to give to the fish and see what results we got. And I know that, I can't remember if we actually did immersion or not, but when we did I know people have also attempted to do the muscular, we didn't get any really good mortalities out of that. So after that, we got a curve, because we only wanted around 20% of mortality with the IP. or just Piscicida. So we did that and we were able to get that through IP. So we kind of just continued with that because bacteria alone, we only wanted around 20% mortality to see if the mortality would increase with the toxin.
Yeah, no, makes sense. Stephen Reichley, who's not on this call today, he tried a lot of different ways to get fish sick with Piscicida, and about the only reliable way was with the IP injections.
Mhm.
So yeah, no, good stuff.
Thank you.
Allie, we have a follow-up question from Dave. He's wondering if you would expect different results with an immersion for the microcystin first.
I would say probably, because when it depends on the trial too. We would have to do an immersion to see how it actually gets into the liver from that point. Because the way that it actually gets into the fish, if it's through an immersion, the amount that the fish uptakes can be very different. So we would have to probably increase the dosage to put into the water rather than inject into the fish. And to be honest, it probably cost a lot because the toxin's expensive. So I would say that yes, we probably need more to get the same results, because we wanted to do a more subacute thing, because we kind of theorized that if the fish are introduced to it, it might be through ingesting the bacteria, the toxin itself. So it could be a lot at once. While I know in a lot of other studies, they always look at like chronic infections with it. But I do think it'd be different.
All right, any other questions out there? Yeah, go ahead.
Hi, great talk. Were you thinking about what would be the mechanism of this increasement in pathogenicity of the bacteria? So is this effect of the toxin on the immunity of the fish? Is this somehow fostering the bacteria growth? What were your thoughts about this?
We think that it is affecting the immunity of the fish, because in fish, the liver is the biggest part of innate immunity within these fish. So I think the fact that it's been able to show to damage the liver, it can definitely affect their immunity, which we think could predispose them to these outbreaks. So I would say, I wouldn't say that it helps the bacteria grow as much. I would think that it's kind of is just damaging the immune system. And then if it's causing anorexia, fish are also going to be weaker in that sense. So.
Thank you.
Thank you.
All right. Well, thanks, Allie, for a great talk.
Thank you.